Okay, in this session, we are going to look at Unit 3 of the practical element of the AS course for CCEA. Okay, the practicals added to the prescribed practicals that you've already done as part of your practical booklet. These are the sort of practicals you can get asked about. Okay, so feel free to pause here and have a look, but it's also on the front of your revision booklet. The first one of these is looking at the food tests. So with each of the food tests, you can see the method there. So you've got reducing sugars, which reduce Benedict solution from its blue color down to an orange or sort of brick red color, depending on the amount of sugars that there are. If it doesn't test positive for reducing sugars, then you would test for non-reducing sugars, okay, which is essentially you treat it with hydrochloric acid to try and break it up, and then a little bit of sodium hydroxide to convert that back to neutral pH or neutralize it. Then again, you just do your Benedict's test. Again, put in a water bath. Again, you're looking for the same color change. So from your blue to green to orange to brick red, depending on the amount of sugars present. Okay. The Benedict's test is an example of a quantitative test. So it gives you, depending on the color, the idea of the quantity of sugar within your sample. Okay. Also, you need to know about proteins. Testing with biuret, which is made up of sodium hydroxide and copper sulfate. Copper sulfate is a light blue color. If tested positive, if proteins are present, then it should go a lilac or purpley color. Starch is using iodine, which is yellow-orange, and it should change to blue-black if starch is present. And then for fats or oils, you use alcohol. You transfer the liquid, so you mix up your sample with the alcohol, transfer the liquid alcohol to some cold water, and you should get what's called a white emulsion, or you should get that spread because the alcohol is absorbed fats. Okay. Also, the next one is looking at chromatography. Okay, so this was used for the chromatography of amino acids. So with this one, it's important that your line is drawn in pencil, okay, so it doesn't run with the amino acids or run with what you're sampling. Also, you need to make sure that the flask has been saturated with the solvent first of all, which helps then your amino acids to run properly. You need to make sure that the line, your pencil line, does not go under the solvent line. It needs to be directly above the solvent line. It should not be at an angle. You shouldn't touch the chromatogram paper with your hands. You should touch the edges, but not the actual main part of the paper, in case something from your hand goes onto the paper. Okay. Once the chromatogram has run, okay, so you have your pencil line, the solvent will run up through and drag the amino acids with it, if you like. Once that has done, then the solvent front, where the solvent gets to, is recorded. Okay, so you mark again on with a pencil, and you'll use that distance when you're finished to calculate your RFs. Also, to help the colors come out of your amino acids, we spray with ninhydrin. Okay, and that can either be done outside or generally in a fume cupboard, because it's harmful, as it says there, when it's breathed in. So sometimes they ask about what substance is used to bring out the color, if you like, in your amino acids. That is ninhydrin. Okay, the type of thing you'll get is this. Okay, and then from that you can calculate what are called RF, or residual factor values. Okay, and the RF always less than 1. Okay, and you generally measure the top and the bottom of your solvent, or your amino acid, sorry. You measure that, divide it by 2, and take the center line. Then you measure that center line down to your origin line, or your pencil line at the bottom, okay, where you've put your amino acids, concentrated spot of amino acids. You take that line and then you divide it by the distance moved by the solvent. Okay, and the solvent should always move more than the amino acids, so it gives you a value of less than 1. Okay, depending on the R groups of the amino acids, more soluble R groups will move further. Larger tends to be larger, less soluble R groups. The amino acid tends to not move just as far from your origin line. Okay. Next bit then is looking at enzymes. Okay, and there's a number of different enzymes experiments that could come up. They may give you some results on this and ask you to draw a graph, possibly, as we'll look at in a second. But there are different things like could be catalase in potatoes or liver. You measure the volume or height of foam. It could be apple puree, which can be broken down by pectinase, and you measure the volume of the apple juice, which is given off. It could be your immobilized enzymes with your alginate beads, which changes color of your clinny sticks. Okay, so showing that glucose is present. Number, as I say, of different experiments they could ask you about this. Again, refer it back to your lock and key or your induced fit. Your could be kinetic energy. It could be the temperature affecting the kinetic energy and then affecting hydrogen bonds if it gets above a certain temperature. It could be pH, which affects ionic bonds, causing the enzyme to denature. You get less substrate enzyme substrate complexes, giving you a lower rate of reaction, etc. Okay, if you are having to draw a graph, remember splat. Okay, so scale is really to do with the scale along the bottom. So you have 2, 4, 6, 8, 10. Each of these spaces needs to equate to the same value of whatever it is at the bottom. You can't go 2, 5, 24, 48. And the same up the side. Also, the scale is to use most of the graph paper. Don't draw a little graph down at the bottom left-hand corner. Okay, try and use most of the graph paper. The points you can plot as dots with a circle around, or X's. Okay, line of best fit, generally with all except the osmosis experiment, is dot to dot. Okay, so the osmosis one is the only one in which you would draw your line of best fit. Axes labeled. Okay, make sure you label your axes with a / for the units. Don't put brackets, put a / and then a title. So it's a title to show generally what goes up the side along what goes, or against what goes along the bottom. Okay, a couple of things there might come up as well. Your homogenization is just breaking up your food sample. So for example, in the food tests, you might put something in your pestle and mortar and mash it up. That's homogenizing or fractionation of the cells. Centrifugation is when you spin them round very fast and you're trying to find their density compared to something else that you know. Okay, so you would have come across centrifugation when talking about Meslison and Stahl's experiment. So things that are light tend to stay at the top. Things that are heavy go to the bottom. Things in the middle are, as you can see, that band in the middle there is in between that density or the heaviness, if you like, of the light and the middle bands. Okay, then you've got a bit about magnification. So MIA, magnification equals the image size over the actual size. That may be done with a scale bar that they could give you along the bottom, as you would have done at GCSE, or it could be that you have, for example, here your chloroplast, and they've marked on a line, and they will have to tell you what the actual size of that line is. You need to obviously bring a ruler with you. Then you would measure either the scale bar or the length of this line, depending on which is, if they have a scale bar, use that one. Then you convert your units. So 1 centimeter is the same as 10 millimeters, which is the same as 10,000 micrometers. If the actual size is 5 micrometers, it'll be 10,000 divided by 5, okay, which would give you times 2,000 as a magnification. Obviously, they could give you the magnification, and then they might ask you to work out the actual size. So you just move around your formula. Okay, so there's an example. Again, pause the screen if you want to take a bit more in-depth look at that. The next bit then is going on to your stage micrometer in terms of trying to estimate the size of the length of cells using your microscope. So you place your stage micrometer on the stage and look through your eyepiece lens. You focus in. This is 1 millimeter, okay, and you can see it's 100 little units here in between, or spaces. 1 millimeter is the same as 1,000 micrometers. So one of these units would then be equal to 10 micrometers. Okay, then you would use your eyepiece lens on your stage micrometer, like so. So here's the stage micrometer, the larger one. Your eyepiece graticule or eyepiece lens that you put in. You can see here that 100 of your eyepiece graticule is equal to 24 of the stage micrometer units. So then you can see that 100 of your eyepiece is equal to 24. You multiply that by 10 because each of these spaces is worth 10 micrometers. Divide that by 100 will give you one small eyepiece unit. Okay, and you can see here that 24 times 10 over 100, each small eyepiece unit, one of those, so from 10 to 11 there you can see, would be 2.4 micrometers. Then you would use your eyepiece to look at cells, be it animal cells, cheek cells, onion cells. You line it up with your eyepiece and you can work out the length of the cell based on this calculation. So if your cell is 10 eyepiece units here, then it'd be 10 times 2.4 would be 24 micrometers in length. Okay, next ones are the osmosis experiments. Again, lots of different types of variations you can do with that. Here on the screen, you can see working at the percentage change. Okay, it tends to give you this type of graph. So here you can see there's an increase in mass or in percentage increase in mass. Better to work out the percentage change in mass because not all the potato cylinders will be the same mass to begin with. So it standardizes your results. In this area here, you've got an increase in mass. So therefore, water is moving in, or more water is moving in than moving out. Okay. Down here, you've got a decrease in mass. This is low solute potential. So that would be a high concentration of sugar or salt solution, and whatever it is. Remember, your zero solute potential is pure water. Okay, so in this area here, we get a decrease in mass. That is more water moving out compared to water moving in. So that the higher water potential would be inside the chip compared to the solution. Going back to this one, higher water potential outside in the solution compared to inside in the chip, so water moves in. At this point, where it passes or crosses the x-axis, that's the point, and the isotonic point, or where the solutions are the same. Okay, so they're the same amount of water moving in as moving out. So the water potential in the solution must be the same as the water potential in the chip. So if they ask you what's the water potential of the chip, then you would go to that crossover point minus 600 and what's that, 30 possibly, that you've got there. Okay, so that's your isotonic point. At that point, that's also your incipient plasmolysis point where the cell membrane is just about to pull away from the cell wall. Okay, in terms of incipient plasmolysis, again, you would do your potato cylinders or your samples in different solutions. You look at them under the microscope and you estimate the amount of plasmolyzed cells compared to non-plasmolyzed cells. And then from that, you can get your results like so. Again, increasing concentration here or decreasing solute potential. The incipient plasmolysis point is where 50% of the cells are plasmolyzed. Okay, so you could draw across your 50%, draw down, and that's the solute potential, excuse me, where 50% of the cells are plasmolyzed, and that's your incipient plasmolysis solute potential. Okay, next one then is looking at the beetroot, or using your beetroot in this case, and then we would use our colorimeter. Okay, so with this one, you can see this is the beetroot one. You have different concentrations, some of which will cause the cells to break or lysis to occur, which will leak out some of the pigment within the beetroot, and the solution goes darker. When you're putting your samples or your vials into the colorimeter works like this. You've got a light source, you've got a filter. The filter you need to use is at the opposite end of the spectrum, okay, to what color your curvette or your solution will go to. Okay, so in this case, if the beetroot is leaking out, it's a red color, which is here, you can see. So we would use a filter that's at the blue end of the spectrum. Okay, it's at the opposite end of the spectrum to your sample color. Okay, your curvette would contain your different solutions with the pigment or with the solution from your beetroot in it. Then you also have a photocell, which gives you a digital readout. Okay, so white light shines through here. If it's a blue filter, that will allow blue light to pass through and absorb the other different types of light. If blue light then hits red light, the red light gets absorbed, the blue light gets absorbed by the red, and you would have 0% transmission or 100% absorbance. Okay, as the solution gets lighter, okay, so let's say at lower potential or lower solute potentials, then more of the blue light will be allowed to pass through. It'll not be absorbed by the red light, so you would get a higher percentage transmission. Okay, so as there's more leakage from the beetroot, the percentage transmission decreases. Okay, and that's the type of thing you would get. There's a type of results there for different, you can see different temperatures. Okay, so the higher the temperature, in this case, there's less transmission, which means then more of the pigment has leaked out. Okay, the next one is looking at the cell different stages of mitosis. Okay, so the important solution really to remember here is acetic orsic stain, acetic porcine stain. They sometimes ask that in questions. So you take your sample of your root, you mash it down, and you use the acetic porcine to stain it so that you can see it under the microscope. Okay, again, it will give you this type of thing. They may give you a diagram, labels A, B, C, and D, ask you to identify the different stages, okay, from that, and you should be able to do that, not too bad. The next one, tissues and organs related to, there you can see your diagram of your ileum. So thinking of your layers here, so your serosa, your muscularis externa with your longitudinal and circular muscle. Then you have your mucosa, or submucosa, sorry. Then you have your muscularis interna or muscularis mucosa, the bits that move the villi. And then you have your mucosa layer, which includes the villi. You could get a block diagram or a plan diagram on that one. Again, when you're drawing the block diagrams, this is from a while ago, you can see it needs to be a block diagram. So you're drawing tissue layers, not cells. Okay, so just layers. It needs to be complete in terms of it needs to have all the different tissues in it. It needs to be accurate and proportional. So it needs to be a true representation of the picture, not something else that you remember. Okay, and also the quality of the diagram. Okay, they don't like these wispy lines that you can see here, or arty-farty sort of lines. If you draw a straight line or a line, it's one continuous line. Okay, no incomplete circles, no overlapping lines that you can see there as well. Okay, then you have the simple respirometer. Okay, so this can be used to work out oxygen uptake. Okay, so you've got potassium hydroxide in here, which absorbs carbon dioxide. Your tap is closed, so you have a certain amount of air within the system up to that bubble. You have your woodlice or respiring tissue in here. It uses up the oxygen, produces carbon dioxide, which gets absorbed by your potassium hydroxide, which causes the amount of gas to reduce within the system, which means that it should move. The bubble here should move to the left, so it should move towards your respiring tissue. You can do that over a time period and then work out a rate of oxygen uptake. Okay, if they're asking for the carbon dioxide uptake, again, you would set it up exactly the same, but you would use water in this case, in which case the water doesn't absorb the carbon dioxide. So again, you would use your respiring tissue, same amount of time. The carbon dioxide is not absorbed, so it stays within that system. If this bubble stays where it is, then you have the same amount of carbon dioxide produced as you had oxygen used. So say going back there, it was what, 40 centimeters cubed. Let's say that's in 10 minutes. Again, if you leave it for 10 minutes, this stays where it is. The amount of carbon dioxide produced would also be 40. Okay. If it moves to the left slightly, there's less carbon dioxide produced. If it moves to the right slightly, then there's more carbon dioxide produced than oxygen. Okay. And sometimes you could get this slightly more complicated looking respirometer. Okay. Hopefully they'll stick with a simple one for AS. Okay. Then we're looking at the heart dissection. They could give you a diagram, ask you to label it. Again, you just need to remember these and learn them. So that's the type of thing they could give you there. External structure and internal structure. Again, pause here and see if you know what each bit is. Okay. You could get some diagrams. For example, there is just a diagram out of the textbook of your arteries and veins. Okay, you might just have to label those, that type of thing. Again, they could give you, ask you for magnification or something there, possibly as well. Then you're getting on to a bit of sampling towards the end. So sampling generally done with quadrats, okay, and that allows you to calculate the percentage cover of plants. So if there is daisies in 4 of the squares, that would count as 4% cover. Okay, it tends to be random sampling, which can be done with random numbers. It can be done like this with measuring tape. You maybe go 20 one way, 17 the other way. That's where you put your quadrat, and you would do maybe 10 in that area, work out your average percentage cover. It could be a bit more systematic, where you have a line set out and then transects off that line, okay, or other measuring tapes off that line. You might measure again at random numbers. You might measure every 2 meters, every meter, or as I say, it could be random intervals as they mentioned here. Okay, so that's the type of thing you can use. Quadrats, percentage cover, good for looking over different types of terrain, or going from, let's say, a forest area to a cut grass area to a set aside longer grass, weeds sort of area. Sampling with quadrats, good for that. Other types of sampling methods are the putter, which you can see at the bottom here, which is looking for small insects. You suck through one end, okay, which tends to have a cover or gauze over it, which means you don't suck it up into your mouth. And then it has another end where you suck up the insects through. You also have your pitfall trap, which tends to just be in the ground somewhere. Okay, leave it overnight, see what insects or what ground hugging organisms that you have. Again, you can make estimations based on that. It needs to be covered so that it keeps out the rain. Okay, if you're sampling trees or rivers or lakes, you would use a sweep net. Okay, or even long grass, you might use a sweep net to try and catch insects to sample those types of areas. What you can do with that is to make a kite diagram. So this would be with the quadrats and looking at a transect. So let's say for bog pine, these are just different species here. There's along your transect. Maybe you take a sample every 2 meters. Okay, so at 0 meters, you can see here that bog pine has a rating of 0% cover. Okay, whereas at 4 meters, if it's got, let's say, 20% cover, there's your 0 line. You go 10% up, 10% down. That gives you 20% cover. You can see these are 50, and 50 makes up your 100%. You can see why it's called a kite diagram, because you get these sort of different shapes that look like kites. And here then you can compare. Okay, so I can see that mountain daisy, there's more of that further into the transect than there is earlier on. It may be that the tussock and the daisies maybe compete with each other here. Okay, so that's why you get less of the tussock here, less of the daisies here. Possibly you can make conclusions based on that. But if it's 100% cover, as you can see with the daisies, after 12 meters, you go plus 50, 50 the other side makes you 100%. Okay, they may get you to draw something like that or answer questions based on, let's say, a graph that they provide. Okay, also in your booklets, you've got this sheet. I think it's an A3 in your booklet. So this goes through each of the methods of the required practicals. So again, your food tests we've been through, the chromatography we've been through, pH graph, you're looking for that type of thing. So optimum temperature, otherwise your ionic bonds get affected, causes enzyme to denature. Your immobilization really is with the alginate beads. So with lactase that breaks down the lactose in the milk into galactose and glucose, which is tested with clinisticks. The colorimeter we've talked about. It may be that you need to do serial dilutions for that, which I didn't mention. That is where you would take 1 centimeter cubed of your solution and add 9 centimeters cubed of water. And that would be it diluted by 10%. Okay, you need to multiply up by 10 at the end if you're doing dilution factors as well. Colorimeter for beetroot, as we've talked about already. Your graticule we've talked about as well. The average water potential is your different solutions, water moving in, water moving out, average water potential there, or the isotonic point as it's sometimes known. The incipient plasmolysis, giving you your 50% value is what you're looking for there. The staining, we've talked about that acetic orcine solution is generally the one you need to remember. You need to know the stages as well. There's your block diagrams for the leaf and the ileum. Leaf is if it's a simple leaf, is pretty easy. You just draw your different tissue layers. Ileum slightly harder. Generally with the block diagrams, there's 4 or 5 marks maybe for the drawing, and then there's maybe another 4 or 5 marks for labeling. So you'll have to remember the labels of each of these as well. Okay, and then the final one was dissecting the heart. Sorry, next to the last one, dissecting the heart, the different parts of the heart that you need to remember, and then sampling techniques we've talked about as well. Okay, hopefully that helps with AS Practical.